So thank you so much, everyone, for having me. My name is Davide Marotta. I'm the program director for in-space biomanufacturing at the ISS National Lab. I'm originally from Sicily, and you heard all my career from Roman. Let's dive into the presentation now. So this morning, I would like to talk to you about space race and the biomedical R&D in space, where it's coming from and how we got here, right? So the first of all, let me try. This is not working. Okay. Um, First of all, let me open up with a quote here. One of my favorite quotes say, Man must rise above Earth to the top of the atmosphere and beyond. Only then will he fully understand the world he lives." So, new discoveries, humanity start to look at Earth from the outside. So the first space race um, started because was like a product of the Cold War, right? So the space race began in 1957, more or less, with Sputnik. And driven power was the political prestige that drove the exploration. Nations competed for dominance in space technology and ideology, and they were just basically like competing to who would put the best tin can in space orbiting Earth. But at the same time, something amazing happened. We started to look at how the human body was behaving in space, right? Some people were claiming, oh, maybe we will turn into a jello because there is no gravity. Some other people say, no, we will survive. But guess what? We didn't turn into jelly. Like we are not like a jellyfish orbiting space. The the human body can endure, but obviously there are some negative effects if we think of bone loss and muscle atrophy, for example. Um, so the biomedical research on human body started as a byproduct of space exploration. And we started to look at the human body with the very first missions like the Project Mercury, the Project Gemini, and the Space Shuttle. So Project Mercury and Gemini were the first project, US project, where they involved the use of human space flights. So that's where the clinician and the scientists start to study and to observe bone and muscle atrophy and the effect on the ocular and vestibular system. The space shuttle from 1981 2011, with a round trip, made space more accessible, and thanks to that, we were able to look and envision the next step of space exploration, which is like, can human being live and work on a constant basis in space? Well, the first project that we had in terms of space station was Skylab and the Mir. So on board of those two first space station, we started to investigate more how the crystals in space behave, and now and how bacteria behave in space, and more studies on the human body. All these was not like planned by the first pioneering scientists who start to work with space. They were just thinking of space in terms of exploration, not space in terms of biomedical R&D platform. They were not expecting nothing of it. The Skylab and the Mir posed the basis for the most incredible thing we have today, is the marvel of engineer, which is the space station. And I'll show you a brief video now where you can see. Let me play. where you can see like the building and the extent of the space station. Not sure if you heard the audio, but the space station is the product of years and years of studies and trip to space is a symbol of cooperation, scientific breakthrough, and I'll give you some spec on these. It's an international collaboration more than different agencies across the world collaborated to the building of the space station like NASA, Roscosmos, ESA, the Canadian Space Agency, and the Japanese Aerospace Exploration Agency. So the space station orbits Earth every 90 minutes, approximately 260 miles above the planet, which should be around 450 kilometers, more or less. Has been continuously inhabited since November 2000 and has been hosting over 260 individuals from more than 20 countries and serves as an orbital outpost and national lab to advance science in space. So the building of the planning to build the ISS started in the 90s, but the realization in the 80s, sorry, but the realization and the uh, assembly of the space station started the very late 90s, and the first humans that set foot on the space station in the year 2000. So it's been 25 years of working in orbit. We do a lot of science on board, and this video is a little speck of what we do. So we have a lot of science on board of the station. Now we're gonna dive deeper into the biomedical side. But so far, thanks to the station, we have observed more than 3,000 experiments on board of the station, five science sent by different researchers in the US and outside the US. So 5,000 plus researchers have sent their payload on station, more than 100 countries. And now we have new countries also sending science to space. We have new players in the space industry. For example, we have Brazil, we have the Kingdom of Saudi Arabia, we have the United Arab Emirates, like players that they didn't have any space program at the beginning, and 4,000 plus than 4,000 scientific publications. We are living today, we are in what we call the decade of results. So back then in 2015, Congress in the United States, looking at the successful story of the ISS, started to think like, okay, we need someone to manage all this science payload on board because only NASA is not possible. And that's they created us, the ISS National Lab, or also known as the Advancement of Science in Space cases. It's a non-profit organization, and we work in cooperative agreement with NASA. What that means that the line, the demarcation line between us and NASA is extremely blurred. Like we blend each other. We work in the same toward the same goals with the same aim. We are in a spatial relationship with NASA. We get money from Congress in order to give grants to researchers in the United States for their research in orbit. The ISS National Lab started, sorry, it's not, okay. The ISS National Lab is focused on applied R&D for microgravity application, new business growth and capital investment using microgravity, leveraging microgravity and the low Earth orbit for scalable and sustainable market and producing a recurring value with the potential to generate demand and revenue from access to space. But now let's dive into the main question that people ask me all the time and ask us, like, why space? Why you want to spend money and time to go to space? So low Earth orbit, or LEO, is more than just the outer space, right? It's the launch pad for innovation where gravity steps aside and let us think differently, work differently, and discover what we couldn't do on Earth. From innovation and discover, from innovation comes discovery, from discovery comes opportunities and new opportunities generate economic opportunities and further advance and driving our work in space. We mainly use on board of the ISS, we can leverage mainly three conditions, let's say: the persistent microgravity, the extreme conditions outside of the station. So think that, as I said before, station is orbiting Earth every 90 minutes. So we have 16 sunrises and 16 sunsets, night and day, in a very short period of time. And that means super hot and super cold in a very short period of time. In addition, you have all the radiations bombarding the outside of the station. So that is the extreme conditions outside, and it's used for extreme testing, materials, thermal cycling, polymer degradation. We also use the station for technology demonstration using the unique vantage points. So imagine you are on board of the station and you're looking at planet Earth from outside. That's your vantage point. That's absolutely necessary for hardware testing, for assembly and orbital services, also like climate change monitoring and oceanographic studies and also archaeology studies. So on board of the ISS, we had so many of those projects. They are almost countless. But then let's look at the persistent microgravity. Thanks to that, we have lack of density gradients, right? We don't have convection motives, like the movement of the air. Convective movement of the air is due because of gravity, so we don't have those. We have a uniform surface vetting. We have a different flow dynamics. We have no sedimentation. We have a reduced interfacial tension. We have a different solidification. And we call microgravity accelerator of conditions. So we use all these, we leverage all those properties that microgravity allow us to test different things for advanced materials, but also for life sciences. For advanced materials, brief introduction, just for you to know what we do also in station, is optical fibers and crystals. I'll go back to the crystals in a second. But also, like, think of new fabric for your clothes. Have you, have a wear dry fit type of fabric when you go to the gym? That was an R&D developed on station. For we harness microgravity for stem cells, regenerative medicine, and bioprinting. But let's dive a little bit deeper. This is not, let me say this is not science fiction. This is science reality. We already bioprinting tissue. We already make drugs in space. Biomedical R&D as never before. Chip technologies, stem cell research. Each one of these fields is undergoing a renaissance in space. Crystals grow more perfectly, more even. Tissues assemble more cleanly. So ways that we cannot replicate on Earth. But why? Why all this in space? So I said before that we don't have sedimentation. Look at crystal branch of our R&D. Convective motive bake crystals in on Earth. Like, how many times we to study protein structure, but these results in a very data because it's still perfect. So this will affect endpoint analysis, where making crystals, thanks to the lack of convective motive, perfect crystals, they form slower than Earth, so they have time to organize in the most stable manner, and you will have a very relation. So the monomers will add by addiction, not by diffusion, because of the lack of convection. And this help us for protein crystallography. We had many projects on board of the station looking at this. And the most famous is RAS oncogene. They were it's a National Institute of Health, NIH study. They unlocked for the very first time the RAS morphology, and they were able to predict and study. They're still studying secondary mutation that will give, for example, drug resistance to crack and decode the morphology for a better prediction in drug discovery. For crystals, compounds drug has been revolutionary because pharma industry, they can reformulate their drugs, and one using space, and one of the most famous experiment and project is from Merck with the anti-cancer Keytruda. Is one of the most powerful anti-cancers. With space, they were able to change, to get better crystals and to change, and they're working for patients with high stability. On the regenerative side, so when you active voltage, when you don't have air movement, your printing is precise. The geometry of your printing is absolutely impeccable because your nozzle, when you 3D print, pretty much is not moving. So these are applicable also on tissues. When you print your cartilage, for example, and your example moment, the nozzle doesn't move. So your cartilage structure is perfect, is geometrically aligned. And also when you inject your cells, the cells will not, because there is no gravity pushing down your cells. So you have an even distribution of your cells, whatever your bioprinting. And for stem cells, we are looking at reprogramming, differentiation, and automation.
David,
Mm-hmm.
excuse me, your connection is not very stable. Is it possible to do something? It's difficult to follow sometimes. I don't know if yeah.
I'm with a Wi-Fi. I don't know what I mean.
Okay, well, okay.
Is it choppy? Sorry.
Yeah, it's
Le-
cut a lot of time during your speech, so that's why.
oh, let me stop my video. Maybe it helps
Okay. Okay, thank
because
you.
that gets a lot of bandwidth from the Wi-Fi.
Okay, okay,
Maybe it's
thank you,
gonna be
David.
better. No,
Okay,
thank you.
thank you.
Let me know if it doesn't work.
Okay, thank you.
So I don't know what you heard now. So well, let's talk about themselves. Some and out. Yeah, like reprogramming on Earth for stem cells is pretty good. The is level cannot get a good view of differentiated cells to use sometimes even in research, but overall for clinical applications. So we are looking at how to improve differentiation in space, but also how to reprogramming in space. Our reprogramming goal is not for the reprogramming itself, but to have stem cells on board of the station to feed the bioprinters, if it makes sense. Because you need to think that when you cells to space is a cost, right? And also we have a limiting factor for the rocket going up and the rocket coming down. So how can we solve these if we need to print tissue or cartilage patients on Earth and send it back? The ideal thing is to send plus patient donor, right? Reprogramming space a good amount of cells for your bioprinting machines, bioprint the tissue that you need, and then send it back. So that's why we are looking at the reprogramming of stem cells in space. So we are like our portfolio is spanning like different products, let's say. But let's zoom in. For example, material inspired Escro Biotech. We have manufacturing of stem cells in orbit. See the sign, and and the bioprinted tissue in space from Wake Forest, for example, liver tissues. But these aren't isolated experiments. They are forming the backbone of the new industry. The in-space biomanufacturing is basically our opportunity to shift from curiosity-driven research to transformational production. Evaluating DNA-inspired nanomaterials to support targeted drug delivery is the first in kind injectable matrix also for osteoarthritis. So they can use this filament for drug delivery, but also to form cartilage. Evaluating the critical steps for stem cell production in microgravity is a path forward in space manufacturing, and evaluating the vascularization is going on board of the station. Another company, Redwire, which is also present in Europe, just bioprinted meniscus. You can see this picture here, very tiny. This is the prototype. Meniscus, but also cardiac tissue. So many people tried to bioprint meniscus on Earth and add cells in the meniscus, the chondrocytes. But there are two problems on Earth. One is the geometry of the 3D product when you buy things. And second is the distribution of the cells. So the cells sink, we have gravity. And due to the lack of perfect geometry, the is not able to support mechanical forces. So it's aesthetically pretty, but when you test the cartilage made on Earth under forces, you smash it. So we don't want this on our knees, right? On knees. Like I actually don't want this meniscus on my knee if I need one. But thanks to space, the geometry is perfect, and we can inject cells, add cells, chondrocytes, and they distribute evenly. They don't sink. So as we say, and tissue engineers say, like it's hard to make hard cartilage that will resist the forces. Instead, we manage these, and Redwire, with a bioprinter facility called BFF, is creating this, is changing the paradigm. Another example is for patient with macular degeneration. So this company, Lambda Vision, it's creating artificial retina, those retinal implants by layer deposition with addition of bacterial rhodopsin. Now, when they create this retina, it's like a right, two stuck on top of each other. On Earth, they never manage to get a perfect product with 200 layers, because if layer number 50 is not aligned, you lose the whole retina implant, right? But in space, thanks to leveraging microgravity, you have the position of those layers with the bacteria integrated. So this implant is ready to go. Different properties of this implant, and then we go into clinical trials very, very soon. An example that I didn't put on those slides this morning, or this afternoon for you, but I'd like to mention are two startups. One is Encapsulate, and the other one is SachoBio. Encapsulate is studying cancer and how to tackle cancer metastatic prediction, and they're using microgravity to as a diagnostic bed for patients. This is the only thing. There are data out there not published yet, but and such biotechnology, you can look at their website that tackles is a new is an RNA based that tackles common pathways in neurodegeneration and cancer. FDA in the United States, they allowed us to start clinical trials, but directly from phase 2 and non-phase 1, because the data are so robust, the data collected in space are so robust, the FDA didn't need the clinical trial, the phase 1 clinical trials, but they're skipping it, and they're going into phase 2. Another project from the Sanford Institute at UC San Diego that I didn't add here, but it's worth to mention, look at ADA1, which is molecular switch for cancer. And they were able to discover a new inhibitor for ADA1 that can act on different different cancer types. And this drug is going into clinical trial phase 1 next month, so this year. These are two successful stories with FDA for new. If you look at those projects, and think what before, microgravity is an accelerator of condition. Everything happens faster in microgravity. So UC San Diego was able to develop and look at the phenotype and discover rebexinib, the new drug, and look at the effect on ADA1 in just 10 days. 10 days in orbit, did several missions, but in 10 days, they were able to see the phenotype, to see the action of the drug, and go to FDA and say, we have a new product, please, let's talk about regulation. So this is accelerating discovery. Microgravity is accelerating discoveries. Myself, I transitioned from the bench and from academia, which was I was tired of the academic world, let's say, to space, because what we are doing in the space industry, in the biomedical, and this is my personal goal and vision, is to recenter the patient at the center of the stage. Obviously, we make money. Obviously, biotech makes money, right? And patients, most of the time, are forgotten. In science, we are focused on, oh, I need to find these and I need to publish and I need to be the first author. And we lose sight of the patient, that they're waiting for us and for our discoveries. So that's my drive, why I wanted to be a scientist at the very beginning of my career, and why I transitioned to space, because I saw a real value proposition for that, a real help for patients, and my advocacy is to put patients again at the center of the stage. And that's what we are observing in the biomedical R&D, space-based biomedical R&D. And here in the United States, Europe, the situation is a bit different. In the United States, we are observing since 2022, since I took this position, the emerging of centers for space research within the different universities. We have new institutions in the East Coast. We have in Boston. We have in Florida. I'm based in Orlando, Florida. We have UCF. We have University of Miami. We have on the West Coast. We have LA. We have San Diego. They're all building those space-based biomedical centers for drug discovery, pretty much. Another topic that I would like to touch with you is organoids, not just brain organoids, organoids in space. Organoids in space. Why 3D models in space? First of all, we don't need a scaffold in space. we the formation of our organoids. I'm trying to be concise here. And if you have questions, please ask later. So microgravity enables formation of larger, more complex 3D structures that better mimics physiological condition of tissues of the human body. Thanks to microgravity, we are able to produce not just organoids like brain organoids or bone marrow organoids, but also to add the immune component and the niche, depend the studies that we need to do, right? So for example, for my, actually I should have, I have a slide on that. So thanks to these, we have more physiologically relevant models for diseases like Alzheimer's, cancer, IBD, improved drug screening and toxicity testing. Also, if we put different organoids on a chip, we can recreate like the body on a chip and do studies for toxicity. I don't know, we put brain, liver, heart, and we test toxicity, drug toxicity. So one, I don't know if you guys are aware, but one thing in the United States is the FDA basically doesn't need animal testing anymore on drugs because thanks to the systems on a chip, we are able to predict the toxic effect of drugs. And actually, some drugs that have been on the market for years and decades, they've been canceled because thanks to the new technology and organoids and tissue on a chips, we were able to identify and discover that those drugs were actually um, um, toxic to cardiac tissue. For example, one famous example is an antihistaminic and an anti-allergy drug, and people were dying with heart attack, and no one understood why. But now we know the correlation. It was highly toxic for the cardiac tissue, and all these were discovered using on chips, but also organoids. So it's not a requirement to do animal testing anymore. So we have an open access and an accelerated path to drug. So, for example, one of the famous projects, it's what I call a helping hand for hair. So the goal was to investigate how microgravity affects human heart function at the cellular and tissue level with a focus of aging, mitochondrial health, and therapeutic intervention. And this was done by Dr. at Stanford University. Another example with brain organoids is this is actually my work, the work I've done before joining the ISS National Lab. So there are two main groups, one in the West Coast, one on the East Coast, by working on degeneration. And thanks to microgravity, we're able to see new pathways. We unlocked, and we are able to see new pathways which are highly druggable. And now those groups are testing and doing that drug discovery for Alzheimer's and Parkinson's. And Sachi Bio, I mentioned before, is using also organoids for their drug testing. But another thing that we need to think in the space industry is standards. How can we protect the community, the scientific community, the space-based scientific community? Because as every new field, space will be full of people making claims that are not true, they're false. Protect. Can we ensure the legitimacy of space-based research, like ISSCR, for example, did with stem cells? If you're familiar with the ISSCR, so we are working to adapt terrestrial standards to the space manufacturing. So what I mean, existing standards for Earth-based manufacturing require adaptation for space application. This will ensure interoperability, enabling different organizations and systems to work seamlessly in space using the same standards. and increasing collaboration across industries and academia. So we are going back to a model where science is highly, highly collaborative, where the community drive the discovery, not the single isolated lab. We are destroying silos because in science, so the modern science closed itself into labs. Like most of the time, the lab group, they don't even know their neighbors. So we are destroying the silos and we are opening up and to the community. So where people can collaborate together. So I created this national network of um, international hubs where people from different universities, different startups, we meet once a month, but we collaborate together and do space-based research. What I mean is, I might be familiar on producing compounds, right? A certain drug, but I have no idea how to make an organoid to test my drug. So the idea is to people talk and combine the different skills. And that's what this is innovation on Earth using space as a platform. One of the important standards also for biomanufacturing is the quality control, the safety and efficiency, providing the guideline to mitigate the risk involved in space-based production. And another one of my other goals is also the workforce development to create a new generation of space-based scientists. They can actively choose their curriculum to do biology in space, biology, microgravity, molecular biology, microgravity, having those curricula that will enable them to be professional in the sector of space-based biomedical R&D. What we are observing today is basically the fourth industrial revolution. So the fourth industrial revolution is driven by AI, machine learning, but also space, because we are harnessing microgravity the same way humanity harnessed different things before, like temperature for metal melting during the Bronze Age, pressure for steam power during the first industrial revolution, vacuum for the digital age, and now we have microgravity that's being removed from our equation to accelerate our discoveries on Earth. So everything we do at the ISS National Lab downward. What is we look at Earth. How can we use space to make Earth a better place? How can we use space to make our society better? And every time a rocket goes up full of science, I get emotional because I know that when the Dragon will come back, the capsule, this will be a better place. This will be a better world with new discoveries, because every time a mission comes back, we have had so many surprises, so many discoveries. Most of them are not published because for intellectual properties, because they will generate drugs. Sorry. So how so much science done in space that people don't even think. So our goal is to make, and my goal is to make this planet better as much as we can using space. And then I put like, this is my book, is published. You can find it on Amazon platform, where it gives a brief introduction of space biomanufacturing for scientists and non-scientists. I wrote this book for everyone really too, because I realized that many people, they don't even know we have a space station, let alone we do science on station, right? So if you're curious about data and results on station, please look our website, issnationallab.org, and you will find the official magazine of the ISS National Lab, um, which is focused on results from R&D taking place on the space station, not just for biology and biomedical, but also advanced materials. You can download for free, and it is a fun read. And then spot the station. If you ever want to see the station, is actually a fun activity. You can spot this. You can use this website to see when the station will pass above you. And believe me, it's incredible. So it and you in the sky super fast, crossing the your sky. And thank you. And that's all for today. And I'm open to some questions